Bonjour, mes amis. It is wonderful to be here with you this evening in our ongoing series where we're exploring nature versus nurture. Tonight, or I guess our last episode, our first installment of this, we looked at how and why it's important not to interpret or view genes exclusively in one-off ways and isolated ways. We explored different pathways. Tonight, what we're going to be exploring is how you have influence over your genes. While your genes may be your blueprint, you're able to dance with them and change how they're expressed through diet, through lifestyle, how you think, what you do. And we're going to be exploring that this evening with our good friend, our colleague, Justin Harris. Um, for those who have their DNA 360, I would encourage you at this point to get it out. We're going to be exploring how you can play and dance with your DNA through four different genes this evening. And so I want to talk to you about what those are and where you can find them before Justin and I dig into our first little topic, which is how we go about changing the expression of our genome. The first gene that you want to be looking at is, or looking up, is APOE. If you have your DNA 360, that would be found in the cardiovascular section. You can look at the summary within that section. The next one we're going to be looking at is COMT. COMT, it's reported a few times throughout the report, but you can look at that one specifically in the mood and behavior section. Again, go into the summary. The next one we're going to be looking at is FTO, and that one will be part of diet and nutrition. And I think if you look at the appetite section, you will find it there. And the last gene that we're going to be exploring is MTHFR. We're looking at two different SNPs. Kind of think about SNPs like plot points in a book, so two different pages of the same gene. Those can be found in your inflammation report, namely the sub-report called methylation. Yeah. So, audience, what we're going to be digging into today is we're going to talk about exactly what is epigenetic regulation, how that influences nature versus nurture. And then Justin and I will be exploring how you can leverage that in your genes that you have, as well as what you can be doing to shift how you're playing with your genes. So, Justin, welcome. Are you excited?
I'm excited. This is one of my favorite topics. This is a full-blown nerd topic. Like it's pretty high level stuff. And it's really interesting, because DNA really is like the hardware of the computer, right? So you can't change the hardware, but
Mhm.
epigenetics, epi is a Greek word that means above. So it's like a more superficial layer of the genome. And epigenetics is the software of the computer, right? So you can download new apps onto your computer. You can mess with the software. You just can't mess with the hardware. But the software has a lot of influence over the day-to-day functioning of the computer, right? So this is something that can be upgraded, updated. You can make it so that it makes the actual system function better. And I love talking about it because I think my favorite part about this is that it puts the power in your hands. Like when we talk about genes, I think a lot of people, it's there's this hesitance because of the doom and gloom side of it. Oh, I have the Alzheimer's gene. I have the obesity gene, right? And it's like, well, no, we need to understand that we're still in the driver's seat here. And that's a really big point
Mhm.
that I want to make tonight. If we have control over our epigenetic information that we're giving our body, these cues from the environment, we can alter that epigenome and ultimately manifest a better phenotypic expression, right? The way your body actually shows up is still very much up to you, folks. So let's have an empowering conversation tonight about how that works.
Yeah, I love it. And audience, please, it's really important to Jess and I that we hear from you. We'll be looking at the results of the poll in a little bit, but we really want you engaged with us. So if you have any questions, don't hesitate to toss them into the question and answer box. If you have something more comments, especially as we're going along, feel free to use the chat. Um, Justin, myself, we make sure we keep that closely monitored. We also have Kate and Vanny who will be helping. And this is Napita. We like to give away things, and so I've decided for the person who can count how many times she comes on camera, because she'll come on and off, at the end we'll randomly pick someone and we're going to send you something fun. So let me know how many times Napita shows up on our screen tonight, and you'll get something fun in the mail. Yeah. All right. So let's dive in. I want to, so we've defined that DNA is your blueprint. And like you said, Justin, there's not really much we can do about that blueprint. But what we can do is looking at the epigenetics. So let's talk about a few ways. Now, audience, there's many. We're going to talk about two, but let's talk about a few different ways that the genome, your blueprint, your DNA can be modified in terms of how it's expressed. What are some ways that the body does that?
Well, one of the primary ways is definitely methylation, right? Like we talk about methylation as being this sort of stickering process. And methylation, it's going on all the time in your cells. It's a whole cycle. And if you guys haven't seen some of the webinars we've done specifically about that, I really encourage you to check those out because we've done real deep dives on the cycle itself and the handful of genes that we look at to determine the efficacy of your cycle versus somebody else's. But the epigenetics come into play here in that I almost look at DNA methylation as like a light dimmer switch, right? So you can think of your genes as being like light bulbs in a gigantic building, right? Your genome being the building in the analogy. And some rooms, like genes, they need to be fully lit, right? Depending on who's in them and the purpose of the room. Some of those need to be dimmed, right? Like not on full blast. And then some of them need to be silenced completely, right? So DNA methylation is almost working like a dimmer switch. And if you add a methyl group, you're turning the light down because methylation actually turns gene expression off. So you're reducing or you're silencing the gene. Or if you remove a methyl group, then you're turning the light back on and you're activating that gene expression, right? And this is happening over and over again in your cells. So just like lights being adjusted constantly throughout the day, depending on who's in the room, DNA methylation, it's continuously adding and removing these stickers as the cell responds to the time of day, signals from food, stress, even emotions, hormones, cell division. So your body is constantly doing this stickering process, and it's really allowing for the way the cells are actually behaving. It's important to remember that every cell in your body has the same DNA, be it your skin, your heart, your brain. It's the same DNA, but the epigenetic regulation channels are what's giving the cell its identity, telling it what to do, how to behave. Obviously, your heart and your skin behave totally differently, but it's the same genome. It's just sort of manifesting differently within the cells, and that's what epigenetics and methylation is ultimately doing.
Hmm. And we, audience for awareness, this is a modification of the genome that we can clinically track to the end of if you send us a DNA sample, we can look at different methylation sites and determine how methylated, how stickered up they are. And then we can also measure those, time period later, 3, 5, 6 months later, and we can see changes in those patterns based on what you're doing. So while Justin, it is true what he said that, those stickers change on a daily basis, when we look at that larger chain, how are we changing the way our genome's expressed, right? That usually takes several months of stickering, but we can quite literally measure that change of stickering, the impact that diet and lifestyle has. So we're not talking in vagaries in literary terms here when we say that you can literally change how your genome is stickered. So keep that in mind. Yeah, no, I know Justin, you love our DNA aging test for this very reason.
Yeah, it's very cool. I mean, they're actually measuring like 950,000 different sites, like locations within the genome. They're called CPG islands. Cytosine, something, guanosine. It's a big, long, word that it's an acronym for. And they're actually able to tell what the percentage of methylation is cell by cell. And then there's all kinds of markers that you can extrapolate from there as to how your body is really working, inflammatory signals, et cetera. It's important to remember, too, that like methylation is not good or bad, right? There's
Mhm.
certain genes that you may want silenced, and there's certain ones that you may want expressed. And the efficiency of that particular mechanism is there to keep you alive. It's also quite the survival mechanism, and it's reacting to everything, right? So your whole life is epigenetic input. The way you eat, do you eat, do you not eat, the way you exercise, even pathogens, toxins, all of these are environmental cues that enter that epigenome, and it has interplay with the genome. And then you get these acquired phenotypic changes based on what you're providing for it. I like to
Mhm.
talk about methylation patterns as well, because it's like, let's say you eat the same breakfast each and every day. Well, that's an epigenetic input that is, in fact, creating a pattern, right? Now your body expects it. All right, 9 a.m. every day, This guy eats eggs and a banana, and that is the epigenetic input, and that's going to cause a trickle down methylation pattern, right? So think about the empowerment of all this. If you have like really bad epigenetic input all the time, junk food, it could be whatever. Think of everything as input that is going to actually change the way that your genes express themselves. If you look at it like that, you realize how much power over your own physiology you have. A simple swap to a higher quality epigenetic input can impact every cell in your body.
Yeah. 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 And I think that becomes really important because I think, Justin, you mentioned that sometimes people get their genes and they're like, shoot, I have the fat gene, for example. So we'll be talking about FTO later. And there's not this awareness that that does not, that influences, but it can be balanced per se epigenetically with diet and lifestyle input. And that's a very empowering, important message because I think science suspected this for a long time, but we're starting to see more and more research come out, as well as big organizations like different cancer societies, World Health Organization. There's some pretty large organizations that are bolstered and founded on scientific research that are really saying, depending on the condition, depending on the disease, 40 to 80% of that is influenced by diet and lifestyle, not just genes. Genes unchecked will 100% influence it, but genes, when balanced with diet and lifestyle, can put it into check 40 to 80% of the time, again, depending on the condition. And we're going to explore that, especially in those four genes that I mentioned at the top of the hour. Yeah. Um, the other quickly, just one of the other epigenetic ways that we can modify things because it's not just methylation. There's something called histone regulation. So, you know how people talk about like your intestines, like your large and your small intestines are a lot longer, but you just kind of cram them all into a space. The same thing happens with DNA, is gloriously long. And one of the ways that we make it a little bit more compact is by wrapping it around these, kind of think about them like balls called histones. We wrap them up. And so, based on how tightly wound or how accessible that cord, the DNA, might be wrapped around the histone, that will also shift how the expression is happening. So, the happy little analogy. Do you remember back before we had little buds in our ears and wireless speakers? We used to have all those cords, all those earbuds are like headphones that would come with long cords. And if you are neat and thoughtful, which is not me, you would neatly wrap that cord up so that it's accessible when you need it. Or if you're a little bit more chaotic like me, you'd shove it in your purse and then it would get tangled. That really kind of think about that being diet and lifestyle influence to a degree as to how we can read histones and how we have access to information. So there's, I'd say probably 10, 12, depending on how you define it, ways we can engage with our DNA. And those are two that become really, really important. So, Justin, talk to me about ways, things that happen in our environment that will shift the way our histones are engaging with our DNA, the way that methylation, acetylation, a few other things are going to be engaging with the DNA. Talk to me about things that we know categorically influence those epigenetic mechanisms.
Yeah, so there's quite a few, right? I mean, I would say there's the introduction modern day of certain things like these forever chemicals is a really good example. You know, these things, our body and our biology are not quite equipped to deal with that particular stressor. And again, if we label stressors like that as an epigenetic input, these things are going to enter the epigenome just like a high quality signal would. And it's going to actually impact the way that the DNA is able to replicate. I think that there's a lot of things modern day, right? Like even Wi-Fi. There's a lot of issues nowadays with mold. I mean, if you think about that, like we take 20,000 breaths a day, a little bit more on average. If each one of them has what we could consider to be a toxin inside of it, and it's going to actually impact the way that the methylation cycle works, because mold actually does damage the methylation cycle, then I think that it's really important to understand that our bodies are reacting to these things 20,000 times a day, right? If you
Mhm.
take 20,000 breaths, each one has something in there that's an epigenetic input that's impacting the body poorly. That's 20,000 inputs a day that are suboptimal. right? So this is the thing, like we've introduced a few things like the forever chemicals, like Wi-Fi, that I don't think our genome is quite set up for, or doesn't quite understand exactly how to interpret or what to do. And I think that it's creating some level of cellular havoc for the lot of us. So that's why I try to put little things in place to deal with these. Like, if you can find, for example, cookware that doesn't have forever chemicals in it, that's a great, just
Mhm.
easy, low hanging fruit thing that you can do. If you can get great, air filters to deal with things like mold, if you can get things to deal with Wi-Fi, like I'm in love with this Aries Tech stuff. I actually have one of these cards right next to my router here, and it's like
Mhm.
it covers like 139 feet, and it actually unravels the Wi-Fi before it can touch my body and therefore enter the epigenome. So little protection mechanisms, right, against some of these modern day inputs that our body's not quite educated yet to deal with. I think that it goes a
Mhm.
pretty long way.
Yeah. 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 No, I agree. And I think we're seeing more and more case specific studies where things like we're seeing evidence of things like forever chemicals in small ways. I don't think we have the computational data yet or computational power yet to understand the how, but we are starting to see the correlation between higher presence of that and more epigenetic stress, meaning stress at certain methylation sites within the genome, right? It's not a one-to-one relationship yet. We still have a bit to get there, but we have a trend within some of our research and data that is suggesting that. Just like, and again, we'll be exploring this in just a second. We have similar data that says like, hey, people with this suboptimal gene presentation, we know that can be balanced with an intervention like this. So it's neat that on different levels, the microscopic versus the macroscopic levels, we're starting to see those shifts and those changes. Before we dive into some of that, Colleen asked the question of can we repeat the genes that we're going to be exploring momentarily and where they are? So, Colleen, as well as everyone else in the audience, look into the chat. Katie or Vanny is going to toss those in just a second. Oh, I think Vanny just did. So, thank you very much, Vanny. Um, and Justin, before we explore those very specific examples, our audience, we've asked some poll questions of. In particular, we want to know what you guys want to learn. So, Justin, if any, Katie, you can toss up the answers on the screen. Let's explore what our audience wants to learn this evening. All right.
Absolutely. We've got it right here. Would you like me to do the honors, Dr. K?
I would love that. Please, sir.
All right. So we asked a handful of questions here. The first one is, how familiar are you with the term epigenetics? 37% said very familiar and understand it well. That's fantastic. 53% said somewhat familiar. I've heard of it, but not confident in your understanding. I'm sure by the end of tonight, you'll have a bit of a better idea. And 10% said not familiar at all. So just from then till now, you've got a little bit of baseline understanding from what we've discussed so far. Question number two, when you hear the phrase editing your genetic blueprint, what do you think it means? 83% of you said that my lifestyle can change how my genes behave. I love our audience with all my
Yeah.
heart, Dr. K. 12% said prevention and lowering risk. Absolutely, that's part of it. 2%, my DNA is fixed and can't be changed. And another 2% said just not sure what it means.
Mm.
Kind of all of the above are of relevance there, honestly, if you think about it.
Mhm.
Number three, we asked, how much impact do you think everyday choices like food, sleep, stress, environment have on switching genes on or off? And a whopping 83% said a lot. They are the main drivers. Beautiful. 15%, some impact, but genetics are stronger. 0% said very little impact. Love it. And 2% said not sure. So we will be diving further into that as we go. The fourth question is, which part of your daily environment do you suspect influences your health the most? And a bit of a mixed bag here in the answers. 37% said diet and nutrition, 41% stress and sleep quality, absolutely. 2% said physical activity. Really? Only two? I'm surprised by that one. All right. And 19% said chemical/toxin exposure. I mean, remember, every one of those answers is in fact an epigenetic input. So, bit of a trick question on our part. It's really all of the above, if you think about it. And the fifth and final question, what would you like to gain most from this session tonight? 48% of you said practical strategies to apply to daily life. Fantastic. 7% examples of gene/environment interactions. 24% actionable tips to apply genetic insights. Just a few percent said information about the testing and interpretation, and 17% said ways to use genetics to personalize health goals. I think we can cover a little bit of all of the above tonight.
Sweet. All right. All right, audience, here we go. Ground, pavement, we're gonna hit it running or something like, well, Justin runs, I don't. I'll skip along. We're good.
Skipping's good for you.
All right. So, Justin, the four genes that we've mentioned, let's explore them. Let's start with FTO. So, what's up with it? If you have a suboptimal presentation, what does that mean? And then let's talk about what you can do or what we know can mitigate that.
Yeah, so FTO, right? The infamous fat and obesity gene, which I
Mhm.
don't love the name, right? Once again, these genes are not correlative in that way. I try not to look at it like this equals this and this equals that. But this gene is of relevance because it has a lot to do with things like satiety signaling in the body, right? Leptin signaling. Um, and if you have the suboptimal allele, which by the way, suboptimal FTO is the A allele, and the T allele is what's optimal. So this really influences appetite and cravings, right? When you start chewing down food, there's actually a signal that goes from the gut to the brain to say like, all right, we're done now. We've had enough. And a suboptimal FTO gene represents a much slower signal, right? So it basically just means that once you stop eating, like your body just got the signal now, and you were actually
Mhm.
done a serving or so ago, right? As far as actual caloric requirement, you were already done there, but the signal just didn't hit you until too late.
Yeah.
And now you've already gone back and you've had the second serving, maybe the third. So a slow gene there, you need to be aware of. It can absolutely influence, BMI and these things, these overeating tendencies that a lot of people
Yeah.
have. So sorry, did you have a
Yeah,
comment there?
there's one other thing too that's I think this thing's really interesting is that this gene or the proteins associated with it are really highly expressed in certain parts of the brain that are associated to reward. Meaning, for people who have a suboptimal presentation here, when they're presented with their favored foods and what this gene will influence favored foods to be, fatty and carby foods, right? So when they see that beautiful croissant in front of them, in their brain, there's actually an activation very similar to pleasure and reward. So they are more strongly attracted to those foods, and their brain has quite literally a greater response to that. So I would offer based on this gene and unchecked, right? We're going to talk about how we check it, but this gene unchecked, people will physically have a greater reaction to the deity that is a croissant or, insert whatever your favorite food is. Clearly mine is croissant.
Yeah, for sure. And that's interestingly, when I'm coaching people, I actually pair FTO with the dopamine pathway. That's just like a
Mm-hmm.
little thing that I do because, like, if you're also in your mood and behavior
Mm-hmm.
section, there's DRD2 and there's COMT, which we're going to talk about a little bit later.
Yeah.
And if you're what we call a high binder of dopamine, right, you get like a pretty intense sensation. That dopaminergic hit that you get is more intense for high binders. So if I see that you're a high binder and suboptimal for FTO, it's
Mm-hmm.
that much more likely that
Yeah.
food may be that vice of choice for you, right?
Yeah.
So this is where we get a lot into the kind of behavioral components. I mean, there's a few things that I recommend right off the bat. I'm always talking about gratitude or prayer or whatever right before your meals because I think that our ancestors, even if they had suboptimal FTO, I mean, this is where the gene started, so who knows? But they didn't have the opportunity to just never ending pile calories down, right? Like nowadays, 2025, we have the opportunity to do it. And I think that it's altered our relationship to the food that we don't have to go through hell to get it, right? Yeah, we got to go to work and get a paycheck and then go to the grocery store, but it's not quite the same reward system that we used to have. So, because of the never ending availability of the calories, I think that we have less respect for the food and less awareness and presence with it. So, part of the reason why I say having a gratitude practice surrounding those meals, it just gives you that presence and that relationship back to the food and be like, I'm aware of
Mhm.
my food. I'm grateful for my food. Take that second before you eat, and it actually does give your body that parasympathetic stimulus so that you can digest it better, and it makes you that little bit more aware of it.
Mhm.
So that's quite useful. I'm also a fan of things like protein prioritization, right? Because it tends to be a pretty satisfying macronutrient. It takes some doing for your body to break it down. So I think that helps as well.
Mhm.
Then there's things like, supplementation with things like Garcinia cambogia. I'm a fan of traditional Chinese medicine. And that one is actually touted as being like an appetite suppressant that actually has some pretty good data behind it. And that's
Mhm.
something that if you are an overeater and you know it, popping some Garcinia can actually help you to sort of moderate that pathway. Um, Go ahead, Dr. K.
I would offer too. So as Justin said, this is unfortunately when we first started exploring, we being the larger community of medicine doctors and scientists, when we first started studying this gene, it was picked up as the fat gene. It became really popular. And so there's been wonderfully because of that, a lot of study into how do you balance it out. And one of the stronger ways to balance it, one of the ways that it's consistently come up in a few studies, if you have this gene, you do have a risk for a higher BMI. You are going to be registering certain foods higher. But if you exercise, audience, 2% of the audience, if you exercise, that quite literally can balance it out, both in from a weight perspective, but also in this is important an experience perspective. Because I think one of the worst things we can, okay, I have a suboptimal presentation of this for those that are asking in the comments. Um, if you have a heterozygous, which is an AT, or a suboptimal presentation with Justin, hold me honest, it's the TT version that's suboptimal. Yep. So if you have
Or wait, with
AT,
that, let me double check. With FTO?
excellent. Yeah.
Okay.
I
I
love that you have a
just checked a
thinking
second
face.
ago. I
Yeah.
swear to
So
God.
if you have,
I like to really double check when it comes to these things.
mm,
Yes, the T is optimal and the A is suboptimal
oh,
for FTO.
okay. So if you
Yeah,
have
yeah.
an AT or an AA presentation, right? If you are registering with that, not only do you want that good BMI, but separate to that, let's be honest, you don't want the mental hell of always craving things. And so some of these studies looked at the proteins and the fibers as well as exercise, balancing that experience out. So, audience, be really mindful. It's important that exercise as well as food choices can not only help with BMI, but balance out the mental experience of it because you don't want to have a great BMI, but be in a mental jail of always wanting more food. So, something that's important.
Yeah, and that's, there's a broader point here that there's a lot of ways to skin a cat, right? There's all manner of input that you can do, everything from the way that you exercise, the way that you supplement, the way that you actually orchestrate your macros. I know Dr. K is cringing because of that saying. I love cats too. Um, there's other supplements as well. Like I'm a fan of ketone esters, right? Like the R13 butane diol. It's got a pretty good appetite suppressant effect. So sometimes I'll stop my meal, right? Like I'll cut it off and then I'll take some ketones or some amino acids or something to make sure that my body's got what it needs, and it helps me to sort of put down the fork right before it's too late.
Yeah.
So there's a lot of little ways that I coach people through this if you do have suboptimal FTO.
Yeah. All right. Let's jump into the next one. APOE. Justin, you want to wrap about what it is and then how we can balance it?
Yeah, so APOE is another one of these things that has one of those blanket terms associated with it, right? Like people call it the Alzheimer's gene, and it's one of those things that we need to navigate that terminology a little bit because APOE, if you have a 4 allele, that is a suboptimal genotype for that particular gene, and it can represent a suboptimal ability of your body to use cholesterol to effectively do its job, right? So when you have inflammation in the body, the liver releases cholesterol to address the inflammation because it's very sticky and Vaseline like, and it sort of helps to smooth out the abrasions that come with inflammation. And it's supposed to be this efficient cycle where it gets released from the liver, it addresses the inflammation, and then it gets pulled back, and everything is hunky dory from there. When you have a 4 allele in the ApoE, what we found is less efficient cycle takes place. So if you've got inflammation in the body and the body releases cholesterol to deal with it, and then there's any sort of toxicity present, the toxins will start to harden that cholesterol, and it can be the buildup of that plaque that causes less and less blood flow to the brain. And less blood flow to the brain, obviously, over time is going to lead to things like neural inflammation. And of course, that can contribute to the Alzheimer's conversation. The theme here that I consider is that we're talking about inflammation and toxicity, right? If we can manage those two things via our epigenetic inputs, then it makes it so that a 4 allele is less and less problematic. We need to make sure that we're encouraging circulation and that we're making it so that the body has less reasons to be inflamed and has less toxin input. Therefore, there's less calcification going on. The more calcified there is, it's like this is a pressurized system that we're talking about, right? So there's going to be less sheer blood flow the more of that plaque that starts to build up. It's like a concrete that sits in there. So what I like to do is I play the game of identify the arsonist, right? It's like who is causing the inflammation, and we can look to the rest of your genetic profile to get some pretty good ideas there, right? Like we can look at your glutathione profile and see, your liver is maybe not functioning that well, or your gut's not detoxing that well. We can even look at your vitamin D profile, right, to see if you're getting that anti-inflammatory benefit of vitamin D supplementation. Another gene there is NOS3, right? If you have really bad nitric oxide genes, that's also going to contribute to less ability of the blood vessels to dilate appropriately, right? So a 4 allele does not mean you're going to get Alzheimer's. I actually have a 3-4, full disclosure.
Mm.
But what do I do about it? Well, I have several detoxification strategies that I utilize on a daily basis, several anti-inflammatory strategies that I use on a daily basis, nitric oxide precursors, sauna, grounding, cold exposure. All of these things together make it so that my 4 can go frig itself, frankly, because I'm not allowing it to manifest as something that's going to cause me harm because I understand how to manage inflammation and toxicity in my body.
Justin, sometimes you dad so hard and I love it. Go frig yourself.
I didn't want to swear, sorry.
No, I value it. I enjoy when you dad out. um, so audience, Justin, what he said there is that having an APOE for allele is not fait accompli, meaning you having it does not make Alzheimer's your destiny. Alzheimer's we know is a multifactorial condition. What does that mean? You can have Alzheimer's without having the APOE4 allele, just like you can have the APOE4 allele and not have Alzheimer's. Okay. But we do know it does increase risk. I don't want to belittle that in this conversation of you have influence over your genes. If you have one copy of the 4 allele, I believe it increases your risk 3 to 4 times. Unfortunately, if you have 2 copies of 4 presentation, in and around 10 to 11 times, I believe. So I don't want to mitigate that. But Justin has just gone over some really important strategies. And I would say working with a clinician to set up a life that is very mindful of how you do this, unfortunately, in a strict way every day, can really go far to mitigate that risk. And so I think that's important for this audience to hear. Unfortunately, it's a very common allele. I believe within the European diaspora, so white folk, I think it's about a 25% prevalence. Within African diaspora, I think it's a little bit lower, 23, 24%. Other populations, it hasn't been studied as well, but we can safely assume 20 to 30% of the world comes up with this presentation. So there's a lot we can do in order to get around it. You're not alone if you have this gene, is what I'm saying. Next, what we want to talk about is going to be MTHFR. This has been popularized as the, to quote Justin, friggin' mother gene. When we talked about earlier in this conversation, methylation, right? Putting those stickers on your DNA. This gene is a really integral part of that. And it is one of the most popular genes that has been studied in this methylation stickering. Okay. Um, and MTHFR, we look at 2 different SNPs within our reports. When I talk about SNPs, think about a gene as being an entire book, an entire story. And when I say SNPs, think about that as two different pages within this book where there's really important plot points, right? You know, when you read a book, some pages are really beautiful and flowery, but they don't contribute to the plot. And then there's pages where you figure out who done it, or you figure out that someone's dead in the library and you see a candlestick nearby, right? So the two pages that we're looking at MTHFR are both reported in your methylation report. And they have different influences. One is going to have more of an influence on cardiovascular outcomes. One is going to have more of an influence on mood and behavior, namely anxiety outcomes. And so, working with a provider, you know, you can really fine tune that. But I want to be very purposeful here. If you have either or both suboptimal, what does this mean? It means that you are not able to methylate folate within that pathway, within that marathon or that relay race, if you will, of getting to the point where you can stick a sticker on, right? These are two really important actors in getting that sticker onto DNA or whatever it might be. Okay. If you are poor in this space of getting that sticker ready, there's a lot of things that you can do to support yourself. Once you know your presentation, you're like, oh, cool. Maybe I need to be supporting my body with different supplements, namely a methylfolate. Maybe folate is vitamin B9. This is what the body, if you have suboptimal presentation here, is struggling with a little bit. Folate comes from the word foliage, which foliage, dark leafy greens. So if you're poor in this, really think of leaning into your dark leafy greens. That can be really, really important. And there's other diet things that we can be compensating with, as well as getting tested in a frequent way. We can look at homocysteine amongst a few other markers in the blood work, methyl methonic acid, if I'm saying that correctly. All of these will give us insight into, are you properly supporting this so that you're mitigating your risk compared to not doing enough to support this? Justin, did I miss anything? I know that the mother frigger gene is one of your favorites.
Yeah, and mostly because it's the mother frigger gene. I mean, let's be honest. It's important to know what the rest of those genes in the pathway look like as well, as far as the what do you do about it kind of a thing. I
Mm,
think most genetic companies, they kind of have a misstep there where they really zoom in on MTHFR. I mean, we've seen it so much. It's one of the most talked about genes. And
mhm,
for us, it's like this is the first step of a pretty complex pathway. And it's like if all the other genes in that pathway are optimal and you have suboptimal MTHFR, you may still be able to have an overall efficient methylation cycle, right? So, I mean, we really look at it from the terms of efficiency and the methylation cycle in its ability to do its job. So we actually look at a bunch of genes. It's not just the one. It is not a one and done thing. There's a whole pathway and cycle. And like I mentioned earlier in the call, it just goes around and around. This stickering process is always happening. And MTHFR is just kind of the first player. It is a very important player, don't get me wrong, but there's more than one moving part. So look at the whole methylation pathway in your reports, and then look at all of
right.
the webinars that we've done just about the cycle, because there's a lot to it. But we have deep dove on it a number of times.
Deep Doven. I like
I don't
that.
think that's a word.
It is now.
Yeah.
Yeah, audience. And remember, I'd really rely on your methylation report to do this deep Doven for you, because it will articulate what form of folate or methyl B9, or sorry, what form of folate or B9, vitamin B9, as well as what form of cobalamin or vitamin B12 is best for you. You don't need to be a geneticist in this space, just look into your report. As well as I'd investigate, especially if you have some suboptimal genes there, talking with your provider, talking with a practitioner of how do I go about monitoring and making sure that I'm balancing this out so that I realize best of health. Because methylation is important for so many different parts of your health. Um, and that beautifully leads us into our next gene. We're going to talk about COMT next. Justin, I wonder, I think this is my second favorite gene. I don't know if we're allowed to have favorite children. I think it's my second favorite child, though. Um, Justin, can you talk about what exactly COMT does and how we can go about modifying that?
Yeah, COMT is Definitely an interesting one. I don't know where it is on my list of favorites, but it's quite interesting, and it really is a methylation gene. You know, like the even the name COMT, COMT, is catechol methyltransferase, right? So it's really got a lot of role in methylation, and it methylates things like neurotransmitters, right? So we talk about it in your mood and behavior report because it methylates epinephrine, adrenaline, right? So that's really relevant for the anxiety pathway. And then it also methylates dopamine. So it makes dopamine available and it also clears it. There's other things like estrogens as well that it's very relevant for. So it's a super important gene within a lot of pathways. And really, what we look at is the speed of it, right? Because if you have like a slower COMT, then it means that you're going to have slower methylation of things like dopamine. So I'll give the alleles here as well. So the GG is fast, the AA is slow, and then the AG is sort of in the middle there, right? So it's good to know what you're looking at there. If you've got really fast COMT, you're going to have fast clearance of different things like norepinephrine, et cetera. So there is ways that you can mess with it as well. Like for example, polyphenols, like you would find in like the green tea catechins and quercetin, those can actually inhibit COMT activity. So if it's too fast, then you can actually slow it down by using things like green tea extract. You can also speed it up, right? So you can actually clear dopamine faster and things like that if you increase methyl donors. I don't recommend doing that willy-nilly, including more magnesium, right? Magnesium is a cofactor as well, or even lowering your estrogen. I mean, via natural means, you can actually do that, and it can actually impact COMT activity as well. It's important to know that in your mood and behavior profile, we don't look at this in and of itself, right? Because we look at COMT and then we pair it with what I mentioned before, the DRD2 dopamine receptor, as well as the ADRA2B noradrenaline receptor. That's how we sort of map out anxiety predispositions, binging predispositions, etc.
Yeah. Yeah. I COMT is so influential because it is that last actor in the methylation pathway. Namely, it's the guy that does the actual sticking. Stickers manufactured, it attaches that sticker. And so the rate at which you do that really influences. And I think you did a good job there, Justin, of articulating that those that are on the faster side of that, the Gregory side of that, they tend to be the warriors of the world, right? Because they're able to power through some of those the stress hormones a little bit quicker. They're able to be more in the moment and rise to the occasion of the stress, right? Um, Conversely, the warriors of the world, those tend to be the AAs, the Andrews of the world. They tend to be a little bit much better with detail, much better with kind of that long-standing work. That said, they don't do well under the pressure and they tend to be a bit more worrying. And so, knowing your presentation there, especially knowing how that's going to predispose towards stress and other different things and how you're engaging with supplements, right? This can really be something where you don't have to be bound by your presentation, your warrior presentation. Knowing it gives you power to better support that, especially in moments where you're kind of facing adversity, right? And so I think knowing that is really, really powerful.
Yeah, and
Yeah.
the AG, right? That medium genotype is interesting too, because that's almost like the best
Mm.
of both worlds. Like it's sort of, we call it more of a Zen genotype. There's a fly buzzing right in my camera. I apologize.
Oh, is
Um,
that what that is?
Yeah, it's
Oh, I
inside,
didn't.
and I'm not sure why, but I'll deal with it after. Apologies, folks. But AG is actually what I have, and so that's good. It's an ability to kind of be zen, and this is the specificity of genetics, right? Because let's say you've already got really slow COMT, and you're just one of these people who's just downing green tea all day long. This is
Mm.
why we talk about it, because on paper, if you say, well, I love green tea, I drink green tea to get the benefits of it, you're also doing some potential downsides, right? You're actually
Mhm.
slowing something that's already slow, right? That's why we love to talk about genetics, because it can even modify some of these little daily habits that you don't think that much about. On paper, it's like, yeah,
Mhm.
I drink green tea, fantastic, good for the body, but it's there's certain things that it can also do for your specific genes that aren't necessarily in your favor.
Yeah. Yeah. And I really like to call, so to the females and the women on the call who have periods, uh, know that COMT is really influential to your cycle. And if you're playing too aggressively with COMT, it will influence your experience of your cycle. And so just something I really encourage women, especially if they think like, oh, I'm going to take anything that Justin just mentioned. It's really important to know the consequence and not overdo it because it will influence your estrogen metabolism, amongst many other things. All right, audience. We're going to start transitioning over into questions and answers because we have a few. But before that, we have just talked about the proverbial fat gene. We've talked about the proverbial Alzheimer's cholesterol gene. We've talked about COMT, which influences stress and how you experience stress. And last we've also talked about MTHFR, the mother Frigger gene, whatever Justin's dadism was. Um, and we talked about a few different ways that you're able to engage with those and either support or mitigate the impact of it. Audience, I would like to know if, especially you have suboptimal presentations there, what you're either doing, current tense, to mitigate that, or now that you've learned about this presentation of yours from your DNA 360, from what we've talked about, what you are going, future tense, to be doing about it. We really want to understand how you're going to be engaging with your genome. So please toss that into the chat so that we know and we can offer cheers and yays, or maybe a little bit of continued advice based on what you're doing. Yeah. And so as the audience is ferociously typing things in, I want to start in on some of the questions that we're getting in the question and answer. Our first question comes from, I hope I'm saying this correctly, Fabiola. And so Fabiola is asking, what is the difference between DNA and epigenetics? Justin, do you want to? I have a thought there. Do you want to offer a thought as well?
Yeah. So your mic cut out just a little bit there, so I'll just repeat just in case. So the question is, what is the difference between DNA and epigenetics? So the analogy that I've made in the past, which I stand by because I think it's fantastic, is that your genes are in fact your blueprint, right? So they're encoded, they're written in pen. You can't erase it. They're going to be there underneath of everything. Like I mentioned, the word epi means above, right? So you've got something written over top of the blueprint, and your cells have to read the blueprint to know what to do, right? So they're going to read that pen at a baseline, but your lifestyle tendencies represent you going over top of that blueprint that's written in pen, and you're writing something new in chalk. Well, that's the superficial layer. It's epi, it's above. So that means that your cells, if they look at that blueprint now, they can only really read the chalk because you wrote the chalk over the pen. The thing about chalk is you can erase it and you can write something new. If you write a message in chalk enough times, it creates a methylation pattern, right? We all know that when we were kids and we were bad kids and we had to write stuff on the chalkboard, the more times you had to do that, even when you erased it, there was still a little mark of that on the chalkboard. And to me, in the analogy, that's a methylation pattern. If you have the same input over and over again, you're writing the same thing in chalk over and over on top, and your cells are reading that chalk. Now, you can still change it, but it takes time. That's why it's a process to change the way your body's methylating things, because different inputs are not going to have an immediate impact throughout your body. But over time, if you introduce different methylation inputs, then yeah, it's going to do exactly that. So that's the difference. Your genes are, like I mentioned right at the beginning, that's your hardware, and then epigenetics are the software. Software can be changed, hardware cannot.
Sweet. Thank you, sir. And you guys can hear me again? Are we good sound-wise?
Yeah, I can hear you.
Okay, fabulous. All right. Our next question is, and Angela, I value, actually it's from Angela and Betty, but I'm gonna borrow Angela's language because I really like it. Justin, what is the doodad called? And that was when you brought up that Wi-Fi doodad. Doodad is a delightful word. Thank you, Angela.
Especially when you're a dad. Doodad is like the best word ever. So the doodad is actually a company called Aries Tech. I didn't really plan on shouting them out. It's just it's right next to me, and it's this is an epigenetic input. So Aries Tech is pretty interesting. They have a lot of third-party testing, and they actually have little bundles and things that you can do, including stickers that you can put right on any Wi-Fi emitting device. And it doesn't block the Wi-Fi such that your devices won't work and things. But what it does is Wi-Fi is like really tightly raveled, right? And it kind of unravels it so that it doesn't have a negative impact on your biology. Even if you have your phone right next to you, but you've got one of these Aries Tech devices on it, it's making it so that it doesn't have the ability to influence your biology negatively. So I use the little sticker things and put them on just any device, but then I've also got the card. I'll hold it up again because it actually is pretty stylish. And I put it right in front of the Wi-Fi extender that I have in my office because it covers like 139 feet. So yeah, Aries Tech. I think it's ARES Tech, and I'm a fan. I think that they've got a lot of cool innovation there.
Thank you. All right. Both Susan and Cindy both Susan and Cindy have our next question. They're asking after MTHFR, namely, what are the suboptimal presentations? So I'll quickly read those out. So for the first SNP, or that first page with a plot point, this SNP is going to be ending in the RSID 1133. It'll be the first one you're seeing in your DNA report. The optimal presentation here is CC, or Charlie Charlie. If you have a T or a Travis, then you will be having a suboptimal presentation. Okay, and this is the one that tends to influence more the cardiovascular side of things. The other one that we look at, the other page, it's the RSID is going to be ending in 1133. It's going to be on the second MTHFR reported in your report. The optimal presentation here is an A, so Andrew. If you have a Charlie or a C, then unfortunately a suboptimal presentation. And so again, you can find both of those in your DNA 360 report. And the second one is the one that's more associated with behavioral outcomes. Okay. Let's see. The next question I have is coming from Philip. And so Philip is saying that they have a suboptimal presentation within both of their MTHFR SNPs, and that they have a few other conditions and a lot of blood work, and they've noticed that different supplements really, really impact their blood work and certain outcomes around these areas, and they're asking just for thoughts and support. Philip, will tell yourself as well as anyone else, this sort of things we can't really answer on the call because we need to ask so many other questions, as well as get a look into the blood work. We wouldn't be doing a service if we answered that question. So if you want to reach out to us via the email ceo@thednacompany.com, we can look at hooking you up with a provider that can support some of that interpretation and offer some counsel there, because they'll be able to jump into a call, see that blood work, amongst other things that are going to be troubling you. Okay, we'll put that email into the chat so that you have access to that. All right, our next question is from Janice. Janice is asking, does the COMT AG, so this is a middle of the road presentation like good old Justin has, does that have any connection with breast cancer risk? Justin, I can take this one unless you have some thoughts. Yeah, I'll take this one.
I think that you might as well go ahead and take it. I mean, I think that AG is typically what I consider to be optimal presentation. That's just my interpretation of it. As far as not slow or fast, kind of in the middle, I've noticed that there's a pattern of pretty good methylation with those kinds of people. But if you're aware of a nuance I'm not, it's all yours.
Yeah, I'll talk through it. And Janice, as well as anyone else who's here, just on the topic of breast cancer, in a month and a half, I believe, October 25, which is a Saturday, we are doing a full day summit. We call it the She Summit. It's another installment of the She Summit, where we are going to be exploring breast cancer. So we are going to be looking at how you can be screening for it, what happens when you have it, aftercare, all things connected to to breast cancer from a genetics perspective. We know that genes are really important to the conversation, but they are not the entire conversation. We're bringing in some really brilliant minds. We have some oncologists coming in, we have a sex therapist coming in, we have people who deal with nutrition, we have people who deal with kind of especially the changing face of it, the younger and younger faces we're seeing. So if this is something that interests you, please ensure that you mark October 25 into your calendar and join us. While a large portion of the conversation will be focused on women, we are going to give a nod to the growing male face of breast cancer, namely that men do have breast cancer or can contract breast cancer, and we don't want to exclude them from this conversation, even though, fair warning, a large portion of it will be dedicated to women. We just want to honor that it's not just women that come forward with this. Yeah, so Jenna, specific to COMT, I have not seen, this does not mean there is not, but I have not seen studies that look exclusively at COMT and breast cancer. This said, there are some correlations because COMT really does influence methylation, and methylation is a really important part of taking out your estrogens. Namely, when I say taking out, when your body is through with them, how do we remove, how do we tag for removal? And so there is some study looking at different methylation pathways, so the full pathway and how that influences it. And I would offer what becomes really important is how you're supporting that pathway in totality. That will influence. I don't think you're going to see a study that says if this gene, then this risk outcome, but we are able to start stepping into the space of if you have this presentation, we know poor methylation can contribute in this way. Here's how we support that. So come to the summit. We'll explore that amongst a few other things. And then if there's a greater concern because of family history, personal history, don't hesitate to book in, and then we can explore how to support that in a little more of a nuanced way. Yeah. And yeah, Janice, I'm glad you're going to keep that in mind. And Katie or Vanny is going to post a link so that people know how to sign up for that summit. Yeah, I'm proud and excited for that summit because I think we have some really great speakers coming. Yeah. All right, Justin, this question is for you. It's along the same vein. It's coming from Sheila, if I can see that far. Sheila's saying if they have a medium COMT, is there something that they should do or other genes to consider? So Justin, when you see someone with medium COMT or any COMT presentation, what other genes are you thinking through when you're approaching how to support the COMT best?
So we have a really important practice called clinical correlation that we do, and I really pay attention to how people are showing up and presenting with their actual health status. Because if I were to just zoom in on COMT in and of itself and just say, well, you got medium COMT, so there's no issues here, you may still be suffering from anxiety, depression, estrogen toxicity. I mean, who knows, right? So none of these things exist in a vacuum. And it's important to know the DNA 360 is absolutely married to the questionnaires and things that we do, the health history, knowing what medications, knowing what your actual issues are, because our job as practitioners is to connect the cellular pathway and the genomic pathway, right? See how things are actually showing up with lab work as well as just clinical presentation. As far as COMT being medium, like I say, to me, that's good because it's not super fast and it's not super slow. It's in the middle. I would also look at things like the rest of your methylation cycle, right? To see, because to Dr. K's point, that's the one that does the actual stickering, but there's a whole cascade that goes on prior to the sticker being put on, right? So there's MTR, MTRR, SHMT1, mother frigger. There's another gene called FUT2 that we look at in that pathway because it has a lot to do with B12 absorption and utilization, which is really important. So I would look at the efficacy of the whole picture there when it comes to methylation. I would also look in your hormone report to see if you've already got like estrogen dominance going on or a predisposition to have like a lot of toxic estrogen metabolites, right? Like 4-hydroxyestrogen. If you've got a lot of red flags within your hormone report, then we also want to kind of pair it with the COMT activity there to get a broader understanding. But at a baseline, I consider AG to be optimal because it's not super fast, which can cause issues, and it's not super slow, which can cause issues. But a real coaching call to really look at the whole works, including clinical correlation, that's the best way to get to the bottom of all that.
Lovely. Yeah, thank you. Um, Susan is asking a question. So Justin, you talked a lot about um, the catechins and other polyphenols that are in green tea, and then that can influence COMT and speed, etc. And they're asking if, with certain COMT presentations, they should not be um, drinking green tea daily. And so, Susan, what I would say is what Justin's getting at is actually more the supplemental side of it. You would need to be drinking, I believe it is about 10 cups of green tea per day to be getting something that would start influencing your genetic presentation. I don't mean that to belittle the benefit of green tea. I think drinking green tea and the polyphenols associated with it spectacular. But typically, what Justin's referring to is the supplementation. You'd be drinking a lot of green tea to get to the point where you're at that supplementation level. Yeah. All right. Uh, Sheila had a question, and this is in regards to MTHFR and which MTHFR. So close. It's been a long day, audience. Which MTHFR influences cardio versus behavioral? I'll just repeat that. So the first SNP that you see in your methylation report, that's the SNP ending in 1133. That is the one that has more influence over cardiovascular outcomes, namely homocysteine. Okay. The second SNP that you see ending in 1131, that is the one that has more impact on neurotransmitter and behavioral impacts. Okay, so the first one you see in your report, 1133, cardiovascular. The second one you're seeing in your report, 1131, that is going to be more behavioral. That's kind of a myopic way of looking at it, but those are the gists of those two pages, those two plot points. Yeah, all right. I think we might have, okay, hold on. Let's see. Our next question is, Justin, I think you might be able to answer this. So we have a question coming in. They're saying that they're contemplating methylene blue, and they were wondering if there's specific genes that you look at to assess for if methylene blue is appropriate. And this question's coming in from Paul, and Paul, I'm going to preface Justin's answer by saying genes alone aren't going to influence whether you should or should not take that. That's the full clinical picture, but Justin could definitely speak to when he's creating that full clinical picture, what genes influence that conversation.
Yeah, and it's really, the piecing together of a toolkit to help with issues is really a lot of what we do. And I think that within the options for that toolkit, there's a lot of different things, right? Lifestyle factors and supplements. As far as supplementing things to help the mitochondria, I mean, the first things that I would do would be more lifestyle related than products, right? Are you exercising? Are you grounding? Are you doing a lot of these kind of basic things for the mitochondria? I mean, if you're not weight training and hitting the sauna and stuff like that, that's always going to be the place to start prior to jumping on supplements like methylene blue. Methylene blue does have some pretty good impact on the electron transport chain. If there's any kind of mitochondrial issue, there's a lot of evidence that would suggest that methylene blue can help you there. And there are genes that really do have some impact there, right? So we talk a lot about the SOD2 pathway. It's more so to do with toxin clearance than actual like mitophagy and things. But if you've got suboptimal SOD2 and GPX pathways, it's pretty likely that your mitochondria are going to get gummed up with free radicals, and it can interfere with mitochondrial performance, right? So that's where some of these other substrates can be useful. Methylene blue also impacts the serotonin pathway. So we also look at genes like 5-HTT-LPR. We look at TPH2. We've also got a whole gut report, right? The Gut 360, which is a fantastic report that you guys should look into because serotonin's made in the gut. And if there's gut dysbiosis, that can impact serotonin. Fixing that might do more for it, right? So if you're looking to use methylene blue for the serotonin side of things, there's a few other things I'd recommend beforehand. But I think it's got fantastically useful benefits for people who have really low energy and who do need a little boost on the electron transport chain. I'm not against it whatsoever. I just think that there's other things I would point you toward first. But those are the genes that I would consider SOD2 and GPX. Did I miss
Well,
anything on that, Dr. K?
no, I think you did a spectacular job there. Yeah. Let's see. The next question I have here is Define. I hope I'm saying that right, Define. They're saying in regards to COMT green tea, does having a regular 1 to 2 cups of green tea or matcha have a significant influence? I'll speak to my awareness of green tea in relation to breast cancer outcomes, and that's a pretty robust set of studies that looks at how much green tea effectively do you need to consume in order to reduce breast cancer outcomes or risk of breast cancer, as well as recurrence. There's a lot of study that has gone into this, and we start to see that needle shift when someone is having 4 to 6 cups of green tea per day. This is green tea, not matcha. I haven't seen comparative studies, but I would speculate because matcha is much more of a concentrated and hit to the system. It might take a little bit less. So that's where we start to see the impact, but that will never be the same as, taking that very concentrated supplement. So I'd say work with a practitioner if this is something you're starting to play with. But for general health benefits, and not specific to preventing breast cancer or something like that, I think 1 to 2 cups, assuming you can tolerate the caffeine, is a really good start. And remember that taking this in, you're not just going to be getting the catchins, if Justin and I are saying that correctly. I don't know how to say that.
Additions? Yeah, something like
Yeah,
that.
there's a whole symphony of other polyphenols that are really important in there. So I think green tea's it's a well-studied, beautiful, delicious agent and thing that you can be doing for your health. Yeah, perfect. And Janice and other people who are still in the webinar, we've just tossed into the chat that detail for the She Summit. It looks like we do not yet have a sign up link, but watch your email for it. Again, it's going to be the 25th from 11 a.m. Eastern, depending on your clock, until about 4. Okay. Let's see here. Janet is asking the question of Janet's asking a question around hydrogen inhalation, if they're worth getting it for a few different health outcomes. Justin, do you want to give a summary on your thoughts of this, and as well as direct Janet to the webinar that we did on this?
Yeah, so we did a very deep dive, like it was like a ingredient spotlight kind of a webinar all about hydrogen. And it was an interesting one because we all had kind of differing takes on it a little bit. It was
Yeah.
Dr. K, myself, and Dr. Varden. And we were all kind of coming at it from a bit of a different approach. Now, personally, I utilize hydrogen. I've never used it in the inhalation format. I've used it in the tablets, right, and drinking it. Mainly to help with the SOD2 pathway, right? So the superoxide dismutase pathway that I have is suboptimal. So it basically means that my mitochondria don't deal with reactive oxygen species that well. They're not going to clear them at an accelerated rate. And because I work out so much, I create a lot of reactive oxygen species. So I have quite a robust toolkit when it comes to dealing with that. Hydrogen is among that toolkit. I know that there is some evidence about inhalation and how it can impact the mitochondria. And really, we talk about different intake methods because breathing something in, it gets into the circulation pretty quick, right? So there's these things called alveoli in your lungs. That's where carbon dioxide and oxygen are exchanged. When you breathe something in, it hits you pretty quick. That's why and this is funny, but if you were to eat a cannabis edible, right, it's going to go through the liver. It's going to create 11-hydroxy metabolite, and it's going to take a long time, and it's going to stay in your system for a long time as well. It'll impact you for hours. Whereas if you were to inhale cannabis, it's going to hit you in about 5 seconds, and in about 45 minutes, it's gone. That's the pathway of inhalation. It's quick, and then it leaves. I think that when it comes to hydrogen, there's some logic to breathing it in because of that, right? The quickness of it, the ability of it to deliver. The research, and I must say this, the research is not entirely hashed out though, right? It's mostly been done in like high level athletes. There is no real research connecting it to Hashimoto's, prediabetes, etc. So right off the bat, I got to say that. I recommend you watch that webinar and really just take it for what it is, because the research is not 100% hashed out.
Beautiful summary. Yeah, thank you, Justin. All right. Alice has a question for you, and this is kind of arcing back to methylene blue, and you mentioned some of the genes that you were looking at, SOD2 and GPX. Can you, for the audience, and Janet, or sorry, Alice, long day, Alice, can you let us know where we can find those in the DNA 360?
Yeah, so there's a big report called the anti-inflammation report, and it kind of breaks down into a bunch of these little sub-reports, a number of which we've talked about tonight, right? So the broader report is anti-inflammation. That's part of the primary 6 reports in the DNA 360. And when you go into it, there's going to be a mini report for glutathionization, a mini report for methylation, another one for vitamin D, and then there's going to be one in there called antioxidation. And that's what we're talking about. SOD2 and GPX, those are antioxidation because they're more specific to the mitochondria and clearing away free radicals. And it's a very interesting pathway. I talk about it a lot on here because I think mitochondria are just huge and very, very important. And when we play Identify the Arsonist, this is one of those things that I look at, because some people have way too many free radicals in their lives.
I love it. Merci, monsieur. All right. Terry has our last question, and audience, we have some goodies after this last question, so you needn't jump off yet. Terry's asking, so close, Terry. Does decaf green tea have the same benefits? And I'll be honest, Terry, I actually don't know, because I don't know the process with which they put the green tea through in order to extract the caffeine, and I don't know if that impacts the polyphenol concentration. So I do not have an answer for you. If you want, I think it's an interesting question, so let me do some research on that. And if you join our Sheet Summit, I'll see if I can have an answer in time for that. So we know if decaf green tea has a similar, it's water process. I just don't, I don't know how that impacts the polyphenols, unfortunately, so I'd have to look into that. But that's a fun botany question, so I will look into that. I like plants. All right, audience. Thank you for being here with us this evening. I have not seen too many comments in regards to if you have a certain gene, what you are going to be doing in order to prevent it. And so, Justin, I wonder if before we sign off, you and I each speak to maybe one thing that we do in terms of if we have a suboptimal gene, what's the one lived habit we have every day in order to balance that habit. Would you like to start, or shall I?
I will start. So I do have suboptimal MC4R, which is actually very similar to FTO. And so I'm a snacker, man. So MC4R, it's the same idea as FTO. I talked about there's a signal that goes from the gut to the brain to say, okay, you're good, stop eating. There's another gene called MC4R that does the same thing, but it's at the level of the palate. So one of the things that I do is I make sure that I chew my food a certain number of times before I swallow it. And it's a very simple tactic. There's other reasons why I do it, because saliva changes the pH of the food and it helps to sort of pre-digest it, quote unquote. But the longer that you can mull around with it in your head before you actually swallow it, it can help to speed up that signal, right? Because you're getting different sensations of the hot sauce and the sea salt and whatever I put in there. So I make sure that I chew the heck out of my food and take my time with it, because that MC4R is not serving me. I mean, I can just not even sense the food if I don't do that. If I'm just pulling it in, I mean, it's all gone and I do not need any more, and I just keep going, right? So I make sure that I chew the heck out of it, and that's a little low-hanging fruit kind of scenario for MC4R.
All right. For me, let's see. I have bad MTHFR. And so I am very careful with my B as in Bob, supplements. For me in particular, that was important because when I look at my whole methylation chain, I'm not someone that should be having methylcobalamin, and yet I was, because I was plant-based for so long. So looking at that report and being very specific as to the supplements that I take into my body became really influential. because I think we all go in with good intentions, right? I was plant-based for 25 years, and I was like, oh, I want the methyl form of B12 because vegetarians, vegans don't get B12, and I want the active form. And that wasn't great for me. And I noticed, I'll be honest, audience, I noticed a marked difference when I stopped taking that. And I can tell when I accidentally take it. I'm a naturopath. I go to conferences and people throw supplements at me, and I'm not always discerning as to which ones I take. And I can always tell when I've accidentally taken a methylcobalamin. And so, as a very low-hanging fruit, for those that don't necessarily have the time and energy right now, at least, to think about chewing or exercising or whatever it might be, making sure the supplements that you're bringing into your body are really fine-tuned. And Justin, I know that you, as the forerunner, the front man of our Black Label supplements, know how important and how much genes can influence what supplements we're taking. So audience, that would be one of my first thoughts. Easy, low-hanging fruit. Don't have to change, just supplement or sub out one pill for another.
Absolutely. Genes can impact supplements. Supplements can impact genes. Supplements can impact other supplements. Food can impact which supplements are necessary. So it's not a willy-nilly thing. You know, when it comes to supplementation, you want some level of specificity there. And that's a big part of what we do with our Black Label.
Yeah. All right. I see in the chat, I've seen Sheila offer how many times she saw the glorious little Nepeta on screen. So Sheila, you are our win-win winner. If you want to email CEO at thednacompany.com with your name and your mailing address, we do need your mailing address to be within the US or Canada. We are able to send you out a very mysterious and glorious prize. So please make sure you email us that so that we can send something to you. All right.
Congratulations.
Yeah, cats always gift. They judge us, but they gift as well. All right, audience. I think that is it for today. We are very grateful that you're here. Yes, thank you, Sheila. I love my cats too. We're very grateful that you've been here. We do have another installation of Nature versus Nurture coming. I believe it is next week. So make sure you stay tuned for that. We're going to explore in particular how AI can influence nature versus nurture. And for those that know, cyborg conversations are amongst my favorite. So we'll be exploring AI's impact to gene interpretation and to your health. If you want replays of this, don't hesitate to reach out. We can support you so that you can see the whole series of this. All right, Justin, any words of wisdom before we dance out?
No, I'm going to get this fly out of my office because it's making me nuts. And I hope you guys learned something. You know, this is always fun. And I'm really glad and grateful that we have the community we have, and it's always growing. And the more you guys show up, the more reason we have to show up. So thanks, everybody, for coming. And Dr. K, you're the best.
Likewise, sir. These are a delight to do with you. I'm very grateful, especially I sit in the clouds and I love how much you're on the ground and you like, Krista, we have to talk about action. Actions is what matters. So thank you, sir. I appreciate you.
Absolutely.
All right, audience, I invite you to do this with us. We are going to be dancing out. We know that exercise influences everything, including mental health. And when we ask the question of what is the exercise that is best for mental health, it is dancing. So we dance out every time. As we dance out, I hope you dance with us, and I hope you have a fantabulous day, evening, morning, depending on where in the world you're calling in from. Au revoir, mes amis. Au revoir.